Alright, so last time we discussed the means through which different types of point mutations within the coding sequences of genes can alter mRNA transcripts and result in the abnormal translation of proteins. We categorized these point mutations according to how they impact the mRNA transcripts and the process of translation. The categories of point mutations that we covered were missense mutations, chain termination mutations, and two subtypes of RNA processing mutations. They were the creation of new splice sites and the eradication of old splice sites. One topic that we have not yet discussed is how these point mutations occur in the first place. In this lecture, we're going to look at the root causes of all mutations and then take a closer look at some specific causes of point mutations. In addition to the classification scheme we looked at in the last lecture, point mutations, or single nucleotide substitutions, can also be classified according to the nature of the substitution of a nitrogenous base. The classification scheme is as follows. A transition mutation involves substituting one purine for another, or one pyrimidine for another. Okay, so here we have adenine and guanine, and thymine and cytosine. All right, and so a transition mutation would involve going from adenine to guanine, or guanine to adenine, or going from thymine to cytosine, or cytosine to thymine. Transversion mutations occur when a purine is substituted for a pyrimidine, or when a pyrimidine is substituted for a purine. All right, so a transition occurs when, for example, thymine is converted to adenine, or adenine is converted to thymine, or when, say, adenine is converted to cytosine, or cytosine is converted to adenine, or when guanine is converted to cytosine, or cytosine is converted to guanine, or when guanine is converted to thymine, or thymine is converted to guanine. In short, the blue arrows represent transition mutations, and the gold arrows represent transversion mutations. It is important to recall that the genetic code is said to be a degenerate code. This property of degeneracy is based in the reality that several different codons can code for a single amino acid. These groups of codons that code for a single type of amino acid are related by their first two nitrogenous bases. For example, all of the 4 codons that code for the amino acid threonine begin with the bases adenine and cytosine. The first 2 bases of those threonine coding codons always reads AC. In total, there are 8 different amino acids that each possess 4 different codons with the same first 2 bases: leucine, valine, serine, proline, threonine, alanine, arginine, and glycine. All of the groups of codons for each of these different amino acids are characterized by a unique combination of bases occupying their first 2 of 3 bases. Because there are 4 codons within each of the aforementioned group of codons that code for a singular amino acid, the final invariable base of each of these codons must include all of the 4 bases present in RNA: uracil, cytosine, adenine, and guanine. Likewise, the third place in the triplets of bases in the coding strand of the DNA molecule that corresponds with these codons must include all of the 4 bases present in DNA: thymine, cytosine, adenine, and guanine. Recall that nitrogenous base thymine in DNA corresponds with the nitrogenous base uracil in RNA. Because of this, if the third base in a triplet of bases in the coding strand of a gene is changed, the triplet bases and its corresponding codon will still code for the same amino acid. Okay? So, if you have a mutation that affects one of the third bases in a codon so that the codon still codes for the same amino acid, you have a mutation that is referred to as a silent mutation. Okay? They do not result in changes in the translated product of the gene. Okay? So in circumstances where a point mutation causes a change in the third base of one of the codons listed above, all right, in thymine, cytosine, adenine, guanine, or I'm sorry, edit that out. That was or in any of the codons of which there are 4 with the same first 2 bases that code for specifically the amino acids leucine, valine, serine, proline, threonine, alanine, arginine, and glycine. If you have both a transversion or a transition in the third base in any of those codons for any of those amino acids, okay, in the groups of codons for which there are 4, okay, that have the same first 2 bases, then you're going to end up with a silent mutation. All right. So here we go. This is an illustration here of what I was talking about. All of these different groups of codons, all right, code for the same or include 4 codons that code for the same amino acid. All right, and these are the different amino acids, okay, which possess at least 4 different codons, okay, all of which within these groups have the same first 2 bases being identical. All right. For example, if you look here at leucine, okay, they all begin with Cu, and valine all begins with Gu, whereas proline, they all begin with Cc. We just said threonine, they all begin with Ac, and then alanine, they all begin with Gc, etc. for these specific groups of codons. So that because the third base in each of these codons could be any of the 4 bases that you find in RNA, then a mutation that affects both the DNA and therefore affects the RNA as well, okay, that changes the identity of these bases either through a transition or a transversion mutation, okay, will not change the identity of the amino acid of which the codon codes for. As a result, you can swipe any of these third bases for any of these groups of codons, and you will end up with a silent mutation. All right. So here you see in the yellow, transition or transversion in third base results in a silent mutation. It should be noted at this point that arginine, leucine, and serine all possess alternate groups of codons. Okay? By alternate, I mean that they also have groups of codons that have different bases for their first 2 bases. These alternate groups of codons have different bases for the first and second places, but still code for the same amino acid. In all of these cases, there are only 2 codons within these alternate groups. Okay? So for arginine, leucine, and serine, you have extra groups of codons, of which there are only 2. They have different bases for their first 2 bases. So speaking of groups that only possess two codons, it is important to keep in mind that 11 amino acids are coded for by codons of which there are only two. Okay? These codon pairs also possess a unique combination of bases occupying the first two out of three bases. For example, histidines, two codons begin with cytosine and adenine. The first two bases of these histidine coding codons always reads CA. All right, so let's find histidine right here. Here it is, right there. All right. However, unlike the groups of codons for which there are four codons for a single amino acid rather, the final and variable bases of these pairs of codons can only be one of two different bases. These two third bases are always purines or always both pyrimidines. So they're always both purines or they're always both pyrimidines. Okay? As a result, silent mutations will only occur in these pairs of codons if there is a transition mutation where one purine is replaced with another purine or one pyrimidine is replaced with another pyrimidine. All right? And by the way, here we have these extra groups here, leucine, serine, and arginine. Okay, remember that they also have groups where there's four codons that all have the same two bases, but then they also have these alternate groups, and I have the alternate groups highlighted in purple. So for any of these three alternate groups or any of these codon pairs that are highlighted in the lighter blue, if you have a transition mutation in the third base, then you will have a silent mutation. But if you have a transversion mutation in the third base, you're going to end up getting a change in the identity of that amino acid. All right? Or you might get a stop codon. As we've seen before, how these different mutations can occur. All right. So again, if a transversion mutation occurs in the third base of these groups of codons, the codons will code for a different amino acid, which would be a missense mutation, or become a stop codon, which is a nonsense mutation. Again, to review missense and nonsense mutations, please see the previous lecture. So now that we have discussed all the specific nature and impact of a variety of point mutations, we will place them in a broader context of all mutations. Mutations can either occur in somatic cells or germ cells. So when you consider mutations, or I'm sorry, rather, when you consider all of the cells of the body, we talked about before how we can break them down into two main categories. You have germ cells in the germline, and then you have somatic cells. All right? So if you remember, we spent a lot of time delineating between somatic cells and germ cells during the introductory lectures. The vast majority of cells in the human body are somatic cells. Okay, these cells differentiate and work cooperatively with one another in all the various tissues of the body. Somatic cells are genetically identical to one another and possess a diploid set of chromosomes. These cells replicate by mitosis to generate two genetically identical daughter cells. All right, and you remember somatic cells because the root word in its soma means body. In contrast, the germ cells belong to a relatively small and isolated cell population relegated to male and female gonads, the testicles and ovaries respectively. Germ cells begin with a diploid set of 46 chromosomes. During the process of meiosis I, all 23 sets or pairs of homologous chromosomes are aligned, and analogous segments of the sister chromatids are swapped between the homologous chromosomes. This event, known as recombination or crossing over, allows for the creation of increased genetic diversity within each of the sister chromatids, of the paired homologous chromosomes. In turn, this results in increased genetic diversity within the haploid gametes that the germline will produce. As meiosis I continues, the pairs of homologous chromosomes separate. When the cells divide, during the reduction division of meiosis I, one of the homologous chromosomes of each set is distributed to each of the two resulting daughter cells. As a result, this diploid germ cell gives rise to two genetically different daughter cells with a haploid set, meaning 23 chromosomes. During the process of meiosis II, each of these daughter cells then separate each of their sister chromatids of each of their 23 chromosomes. Meiosis II is completed when one sister chromatid from each of the 23 chromosomes of the original daughter cell is distributed to one of the two new daughter cells resulting from the second cell division. This second division occurs in each of the original daughter cells that resulted from meiosis I. As a result, meiosis II gives rise to a total of 4 haploid cells. In the male germline, all 4 of these haploid cells gives rise to sperm. In the female germline, only one of the 4 haploid cells resulting from meiosis II is destined to become the ovum or egg. Both sperm of the male and the ova of the female are collectively referred to as gametes. If any of this preceding information is confusing or unfamiliar, please refer back to the Chapter 2 notes. This is all review. Mutations that occur in somatic cells are known as somatic mutations, and mutations that occur in the germline are referred to as germline or germinal mutations. All right? So again, mutations that occur in somatic cells are known as somatic mutations. Recall that these cells replicate by mitosis to generate 2 genetically identical daughter cells. Therefore, if a mutation, symbolized by the blue cell, occurs within a single somatic cell, it will be passed on to the progeny of only that specific cell. All right, and it won't be distributed throughout all of the cells of all the tissues. Mutations that occur within the germ cells are known as germline or germinal mutations. In meiosis, germ cells undergo a complex set of genetic alterations as they divide to yield gametes. The timing of the process is different in the male and the female. A germline mutation can occur anywhere along this complex series of divisions. If a gamete impacted by a germline mutation is incorporated into a zygote, it will be distributed throughout all the somatic cells that result from that zygote. As a result, a germline mutation can permanently change the genome of every cell in a person's body. This same mutation will also be incorporated into the germline of the person that develops from the affected zygote. As a result, germline mutations can be passed down through the progeny of an affected individual. In contrast, somatic mutations are relegated to a limited cell population in the individual from which they arrived. This is not to say that somatic mutations are less harmful than germline mutations. For example, somatic mutations that change the functioning of genes that regulate cell maturation and division can result in cancerous cells that replicate out of check and cause pathophysiological havoc. In addition to classifying mutations according to the type of cell they originate in, mutations can also be classified according to their root cause. In general, the causes of mutations can be broken down into 2 broad categories: spontaneous versus induced. Spontaneous mutations are thought to occur without any environmental trigger. They can be the result of chemical changes and reactions within the DNA that tend to occur on their own. Such mutations will result in point mutations that we have already discussed. Spontaneous mutations can also result from errors in the process of DNA replication and recombination. These types of spontaneous mutations will result in deletions and insertions. Deletions occur when small or large segments of DNA are eliminated from their normal location within the DNA molecule. Insertions occur when small segments of DNA are inserted, or small and large rather, segments of DNA are inserted within pre-existing sequences of DNA. Induced mutations are mutations that are caused or influenced by some environmental factor. Any factor that causes a mutation to occur or influences the rate of a mutation is called a mutagen. A mutagen can bring about mutations in four main ways: electromagnetic radiation, chemical agents, viruses, and transposable elements. With regard to electromagnetic radiation, short wavelength or higher energy radiation such as X-rays and gamma rays tends to bring about the most damage to DNA. And X-rays and gamma rays, this high energy electromagnetic radiation is like, it's almost like, a motorcycle accident for your DNA. It can break it in all kinds of crazy ways. We're not going to really go through all the different ways in which that can destroy DNA, but suffice it to say, it's very effective. All right. Chemical agents can react with nitrogenous bases to cause a variety of point mutations, which we're going to talk about soon. Certain viruses possess the ability to insert their own DNA within the DNA of the human genome. This viral DNA can in turn disrupt the coding sequences and regulatory elements of genes. Finally, transposable elements are a special subtype of repetitive DNA sequences that are located thousands of times interspersed throughout the genome. These repetitive DNA sequences are found repeating themselves thousands of times throughout the genome because they represent segments of DNA that were once able to jump from one area of the genome to another. The transposable elements are interspersed repetitive sequences that continue to maintain the ability to jump from one area of the genome to another. The most common of these jumping genes are subtypes within the line and ALU families of interspersed repetitive sequences. When these transposable elements jump from one part of the genome to another with the help of certain transposable enzymes, they can result in disease-causing mutations landing in the middle of the coding sequences and regulatory elements of genes. Now, from reading and reviewing the basic classification schema of mutations previously presented, you may recognize that point mutations can be both classified as spontaneous mutations or induced mutations. Another way to think about it is that both induced and spontaneous mutations can take on the form of point mutations. For the rest of this lecture, we are going to look at how the point mutations, initially discussed in Mutations 1, can be created spontaneously or induced by a mutagen. During the Mutations 3 lecture, we will take a closer look at the spontaneous mutations, further classified as deletions and insertions. And by the way, I should say that spontaneous and induced mutations that can be classified as deletions and insertions. Spontaneous point mutations occur when nitrogenous bases rarely spontaneously shift to another isomeric form. Recall from general chemistry that an isomer of a molecule possesses the same number and proportions of atoms and thus has the same chemical formula, but possesses different connectivity between the atoms. In nitrogenous bases, this shift, known as the Tautomeric shift, involves movement of a hydrogen atom. In the case of both guanine and thymine, hydrogen is usually covalently bonded to one of the nitrogens within a ring of the base. This arrangement occurs in the keto isomer. In a tautomeric shift, this hydrogen can be shifted so that it's connected to one of the oxygens covalently bonded to the ring, okay, to form what is known as the enol form. In the cases of both adenine and cytosine, they usually are configured so that they possess an amino group. This configuration is known as the amino isoform. However, a hydrogen atom of this amino group can shift so that it is then covalently bonded to a nitrogen within a ring of the base. This configuration is known as the imino form. Okay, so the tautomeric shifting of the keto form, all right, of guanine and thymine, okay, to the enol form is a possibility, or we can have the tautomeric shift of the amino form of cytosine and adenine, okay, shifting those two bases towards their imino form. So what's the relevance of this? In all of the above-mentioned scenarios, the tautomeric shift of these bases will result in improper base pairing during DNA replication. So during DNA replication, the presence of the alternate isomers of these bases results in the wrong purine being base paired with its respective pyrimidine, and then vice versa. Okay? So here we see incorrect complementary base pairing with enol isomer of thymine results in thymine being erroneously base paired with guanine. And then the incorrect complementary base pairing with amino isomer of cytosine, okay, results in incorrect complementary base pairing with adenine. All right. And notice both guanine and adenine are in their normal keto and amino isomeric forms. So after replication, the shifted base on the parent strand will shift back to the more common isomer. So these guys are flipping back and forth. That's why it's called a tautomeric shift. As a result, the parent strand will not be altered. However, the mismatched base pairing will result in the wrong base being inserted within the newly synthesized strand. Okay, so you got to think about the semi-conservative nature of DNA replication. So a thymine base undergoes a tautomeric shift prior to DNA replication. All right, so here we have the base mismatch being carried over to the newly synthesized strands, okay, which are symbolized with the blue, as opposed to the older strands, the parent strands, being symbolized with the color purple. All right, now that mutation is embedded, you can see right here where once we had adenine, now we have the erroneous mismatch of guanine. That erroneous mismatch is now going to be carried on to subsequent generations of cells, okay, that happened to have inherited that error. All right. So when this newly synthesized strand acts as a parent strand, this one right here acts as a parent strand for further replication cycles, a new point mutation will be permanently embedded in that lineage of cells. All right. Induced point mutations can occur by a variety of chemical agents. We are just going to visit one of these agents and the mechanism by which it works. However, keep in mind that there are many mutagens that work in a similar fashion. One of the most common mutagens to which we are regularly exposed is nitrous acid. Nitrous acid is often found in its polyatomic ion form within the salt sodium nitrite. Nitrite is the polyatomic ion that results when nitrous acid donates its proton. Sodium nitrite is a very common food preservative. It is especially common and plentiful within processed meat products. Next time you go to the grocery store, look at the ingredients and preservative lists for your processed meats. You will find that most of them are preserved with sodium nitrite. It may be rare that you might be able to find processed meat products without sodium nitrite, but usually those products are also more expensively priced. This is because sodium nitrite is an extremely inexpensive and effective preservative. The fact that it is a salt containing sodium also adds to the taste of food. So in your GI tract, the nitrous acid dissociates into salt and nitrite, and the nitrite then, the sodium nitrite dissociates into sodium and nitrite in your GI tract, and the nitrite then readily recombines with protons to form nitrous acid. Nitrous acid participates in a chemical reaction known as deamination with the nitrogenous bases cytosine and adenine. So here we have cytosine and adenine right here for us. In deamination, the amino group of cytosine and adenine are replaced with ketone groups. All right. So here we can see the amino group being replaced with a ketone group. And then in adenine, again, this amino group is being replaced with a ketone group as well. Well, this changes the identity of the nitrogenous bases. In adenine, this will result in a new nitrogenous base that you may have never heard of before, hypoxanthine. In cytosine, this results in the formation of the nitrogenous base uracil. Yeah, uracil in DNA. All right, that's against the rules, right? When replication of the DNA is initiated before a cell divides, the change in the nitrogenous bases cytosine and uracil, the change in the nitrogenous bases from cytosine to uracil or adenine to hypoxanthine in the parent strand will result in the incorrect complementary base pairing on the newly synthesized strand. More specifically, the uracil that was previously cytosine will form a complementary base pair with the adenine on the newly synthesized strand, and the hypoxanthine that was previously adenine will form a complementary base pair with cytosine on the newly synthesized strand. As a result, the newly synthesized strands will give way to future DNA molecules in future cells that possess a point mutation that is located at the same place as the nitrogenous base that was originally deaminated by the nitrous acid. All right, so it's basically the same concept as before, is once you alter the identity or nature of the one of the nitrogenous bases, then you are going to erroneously base pair that nitrogenous base on the newly synthesized strand during DNA replication, and then that newly synthesized strand, which now contains an erroneous nitrogenous base, okay, will now be carried on, okay, when that newly synthesized strand acts as a parent strand, all right, and it will just be carried down on and on and on and on and on, okay, throughout all of the cells, which unfortunately derived upon that cell where the mutation originally was created. Okay, and this can happen either in a somatic cell, okay, or it can happen along the germline. All right, so I think that does it for now, and stay tuned for Mutations 3.